Hello and welcome to Ask Your Academic. Today's session will cover the MSc Medical Genetics and Genomics, and we're joined by our programme lead. He's here to answer any questions you may have about the programme. Some questions have been submitted before today, so we'll do our best to get through all of those. You can also use the chat box to ask any other questions you might have, and you can also use that to say hello and let us know where you're watching from. I'll pop some helpful links into the chat box too for you. If there's anything sort of specific about your application or funding or that, that'll be a useful place to look. If after the session you have any further questions, you can contact the programme administration team, and you'll find the email address for that on the programme webpage on our website near the top in the blue box. We are recording this session, so that will be made available to you shortly afterwards to watch anything back that you'd like to see. So we do only have a short session, so I'll get our panel to introduce themselves and give a quick overview of the programme, if that's all right.
Okay, good morning, everyone. Thanks for joining us. My name is Gerhard May, and I am the Program Director for the MSc in Medical Genetics and Genomics. Bit about my background. I'm actually not British. I'm from Germany. From South Germany, but I moved to Scotland in 1990, so most of you will not have been born by the time. So I've been in Scotland for a long time, but what's relevant here is that I know what it's like to move to a different country and to talk to people who I don't understand because they speak in a funny accent. So I think that's an important aspect here, that I know how you will feel. So the MSc in Medical Genetics and Genomics. Oh, a bit more about my background, actually. I'm by training a bacterial geneticist. So I've nothing to do with medical genetics. I've learned all of this on the job. And that is probably also an important aspect. So whatever you do in your master's program does not necessarily tie you into a given career structure that from now on you will only ever work in medical genetics. It will give you skills that are transferable and allow you to move from one field to a different one. So I've joined the Medical Genetics and Genomics team at the Glasgow University in 2016. And I've been the director of this team or the program for the last 2 years. So this is my second academic year that I'm the director here. We're quite a small team. There's only 6 academic members of staff who teach on the program, so university employees, and we're lucky we have one teaching technician who supervises lab work. So to show you a bit about the Medical Genetics and Genomics program, I have made a PowerPoint slide here. I hope you can see that now. So this shows you take the laser pointer, shows you the months here of the academic year. Like all master's programs in the UK, this is a 1-year program, and you will be studying courses that add up to a total of 180 credits in the Scottish higher education system. Now, the courses I've highlighted here in colored boxes. Before Christmas, so that's New Year here, before Christmas you're studying one single course, which is called Genetic Disease, is worth 1/3 of the total credits of the program. And this course consists of lectures taught by academic staff from the university, but also a significant number taught by members, employees of the National Health Service in Scotland. So these are people who work in healthcare and come and help us and teach you guys. There's not only lectures, there's also small group work, tutorials. There is a bit of lab work, but we'll get back to that later on. In the second semester, that's January to March, you will be taking 3 courses. 2 of them are compulsory. That's the orange one here, Case Investigations, a 20-credit course where you will be working in groups, and we'll get back to group work in a short while as well. The other one is called the SNP assay design. This is the lab-based mini project where you will be designing a new genotyping assay, and you are going to plan your own time. There is another course here in the second half of the second semester, which I just put down as option. This is the only place or time point within the year where you guys will have a choice. There are 3 different courses that are available to MSc Medical Genetics and Genomics students. One is called Clinical Genomics, and that is taught by Professor Ed Tobias. He's one of our teaching team. The other 2 courses, Next Generation Sequencing Bioinformatics and Cancer Microenvironment, are taught by staff from other schools, but there are always places available for students from our program who are interested. In the 3rd semester, so from after the Easter break, you'll be studying one short 10-credit course that's only a couple of weeks long on disease screening. This is where you will learn what a screening program is, how to evaluate these. And then from about middle of May, so it's actually started just 2 weeks ago, from middle of May, you will be working on the final master's research project or dissertation. This is also worth 60 credits. So that is the second big chunk of the master's program. So our students, the current academic year, they just started on their project Monday last week. And I think it's going okay. I've met some of them yesterday evening at a barbecue, and they seemed to be okay, but I don't know if they would admit to me if they hate it. Any other questions about that program in general?
So we actually had a few questions asking more about the project. So in terms of how students choose their topic, their supervisor.
Okay, the project. This is a difficult topic. There are a number of different types of projects that are available. One is a wet lab-based project type, where you'll be joining an existing research group, for example, or where you'll be working together with other students of your or other master's programs on a sort of group parallel project. But that will all lab-based, and you'll be writing up a research report on what you have found out. A second type of project is what we call dry projects, and they are usually or should be data analysis type projects. Often they are bioinformatics related, so often what you'll be asked to do, you'll be given very large, potentially very large data sets, which you are then expected to analyze using bioinformatics tools. Now, this is something which you will then learn while doing the project. There are also usually projects that are offered by members of the National Health Service. So often we have what we call audit projects, where students get access to, I don't know, 3, 4, 5 years worth of patient data, and are expected to evaluate how introduction, for example, introduction of a new diagnostic test, how that has affected the patient journey and the diagnostic rates. A third type of project is what we call a systematic review. So this is also a research project. So it's not simply reading papers and writing a review about it. It is identifying systematically all relevant publications for a particular topic. So there is always a research question associated with that. How are the projects chosen? Students are given a very large list of projects. I think this year in our college, in our cluster, this master's program is part of a cluster of biomolecular master's programs together with biotechnology, biomedical sciences, and some others. Students in this cluster will get a very large list of about, this year was about 130, 140 different projects, different supervisors, and each student is then asked to choose their 5 top choices and choose keywords, specific topics that they are particularly interested in, cell signaling, cancer, diagnostics, or also technologies that they would like to learn, techniques. Then an algorithm is used to try and allocate as many students as possible to a project of their choice. Now, this year, this algorithm was actually quite successful and managed to allocate about 65 to 70% of students to one of their 5 chosen topics. But you need to be realistic. There are also always some students who don't get allocated to a project of their choice. In almost all cases, we were able to identify projects that at least corresponded to some or most of the keywords that students indicated, so key techniques or keywords. But it has been a struggle. This current academic year, we have a very large cohort, not only our class, but also other master's classes. It has been difficult, but the university is aware of this, and I'm quietly optimistic that next year, when it gets to you choosing projects, this is a bit easier.
Perfect. Excuse me, thank you. And whereabouts are the classes located? Are you on main campus for most of those?
Okay, I'll show you my, because I was told before that these questions would come up, I prepared some pictures. So this is a Google map screenshot of Glasgow. And here you can see the city center. George Square is where the city chambers are. These are here and here are 2 of the main railway stations. So that's where the city center is. This bit here is where the university campus is. So this is called the West End. This is a main road called Byers Road, where all student life happens. And the university itself is in this gray block here. Currently, all our teaching is on the main campus. However, our team, and this is going to be a bit a longer story now, our team is actually based over here. This is on the south side of the River Clyde. This is a major hospital. It's the biggest hospital in Scotland, and it's called the Queen Elizabeth University Hospital. So that's where we are based. There is also a dedicated building there called the Teaching and Learning Center. So for many years, all our teaching, all of it, happened in the Queen Elizabeth University Hospital Teaching and Learning Center. However, with the outbreak of the pandemic, that building was handed over to the Lighthouse Labs, and it was used for the testing facilities. So for all the PCRs in Scotland basically were done in that. I'm not sure, that's probably exaggerated, but the large number of them was, there were, at the height of the pandemic, there were 600 people employed in that building. But that means this building is still not usable as a teaching facility because it had to be completely refurbished. Now, we have been assured that from the next academic year, all our teaching will again be happening in the Queen Elizabeth University Hospital. But I personally don't trust that yet. So I think our timetabling experts at the moment actually timetable everything in double. we're timetabled down here, but also on campus, should we not be able to move everything to the hospital. Now, what I also wanted to show you here, these gray areas are where, to my knowledge, the main university student accommodation areas are. So this is the university owns accommodation, and this is where some of these places are located. They are, of course, very much more convenient for the main campus compared to the hospital. But here is a train station called Partick, and from there are buses that go directly to the Queen Elizabeth Hospital every 10 minutes or less than that. So I think it's even more than 10 minutes. Yeah. So getting to and from the hospital is not a major issue. It is, well, some students live in the center, and it's a longer trek out to the hospital from there. But we're expecting to move most of our teaching back to the hospital.
Perfect. And I'm assuming that'll all be confirmed once students are enrolled and they've picked their classes, that'll all be kind of communicated to them where that'll be.
Well, yes, I should, as I said, there is really very little choice in your program. So you will be, should you choose to come to this master's, you will be enrolled in all the compulsory programs. You will have basically not to do much choosing there, and then you will be notified of where these classes are. You'll be given timetables. There is also an app that can be downloaded onto your phones, which is called UofG Live, which gives you an online timetable, which we try our best to keep updated so that students always know where they are supposed to be at what time.
Perfect. And there's a question came through our chat box asking how many hours a day are you typically in class, if there is anything such as a typical day?
I'd say on average, maybe 2 to 3 hours per day. I'd have to look this up actually and calculate this because we are planning for the teaching to be at the Teaching and Learning Center at the hospital. And we know that students really don't like traveling there for just an hour, one lecture or something. We are going to do our best to timetable everything relatively compactly so that it's maybe only 3, ideally, well, certainly not more than 4 days a week that you would have actually in-person classes in the hospital. You'd have, so we're going to try to keep a day free for personal study. I should say there are specific expectations associated with what a university credit actually means. So 1 credit is meant to be approximately 10 hours of learning. So for a 60 credit course, it works out to about 40 hours per week that you should expect to spend about 40 hours per week in class or in personal study. And we're getting back to that with a different question. This is going to be a full timetable, and there is an expectation that you're spending a significant amount of time in your own time working. I should also say, we all learned during the pandemic that we don't have to do everything in person in a classroom. The use of Zoom or Teams or something like this is going to be here to stay. So there is certainly, we will be doing some things still online because it's just more convenient for everybody. But we're planning to do the majority of our lecturing in person.
Perfect.
Does it answer the question? Yeah, I did say.
Yeah. And is there, I think you did mention it, but you didn't go into much detail. Is there any group work or lab work on this program?
Yes, so as I said, when I showed you the timeline, this course SNP assay design and validation, it's actually called, long name, that is a mini project. There's one introductory lecture, and then you'll be working on your own project where you'll be in the lab whenever it's convenient for you. Because you're studying other stuff at the same time, an optional course, you can't be in the lab all the time, but it's entirely up to you to timetable, to schedule your time in such a way that you get your lab work done, and it's in your interest to get it done as quickly as possible. So that's a mini project, and it is actually a research project because you'll be given a single nucleotide polymorphism, so that is a location in the genome where 2 or possibly 3 or 4 different alleles are present and are possible within a population, and you are expected to design a completely new novel genotyping assay based on PCR. In the first semester, there's also a little bit of lab work. There is a practical, bit more like an undergraduate practical, where we know what the answer is, and you will come in and work mostly so that you all get the hang of using the equipment in a lab. Because let's face it, not every, especially during the pandemic, not everyone had a chance to do lots of lab work. So we always have the experience that there are students in our cohort who don't really know how a pipette works, and have not very strong skills in this, often quite nervous students. And if you fall into that category, so don't be nervous, because we'll help you. We'll try to help you to gain these skills so that you actually are able to work independently. So a bit of lab work in the first semester, a significant mini project in the second semester. Group work. There are a significant amount of our teaching and learning works in groups. So students in the first semester and in that case investigations course in the second semester will be put into groups. In the first semester, you'll be, and these are groups of about 9, maximally 10 students. And in these groups, you'll be meeting at least once a week, and you'll be allocated cases. Some of them are actually based on real life cases where you are given information. You are expected to figure out what kind of diagnostic test one would apply. You then would request diagnostic tests. You will be given the results. You're expected to interpret them, and you write little report of them. Now, there is not only individual report writing, but also group work in group report writing and presentations involved in this. So yes, prepare yourself mentally. You will have to work in teams with other students and do work with them together. That's a really important skill, okay? I know that there are always students who don't like that, but real life, and you can ask anybody, real life consists of working in teams. You have to work with people, even if you might not like them, you have to work with them. And that's something that is really important to learn.
Yeah, thank you. And someone asked, does the programme include workshops in case students are needing some revision or a better understanding of the subjects covered?
Yes, I'll get back to that in a minute as well. We have a preparatory course on our virtual learning environment, so students can prepare themselves a bit, and get back to that question in a second. But during the academic year, certainly during the first and second semester, what we do is we allocate each student to a member of staff as their mentor. So we'll try to keep track of how students are doing, and students who are struggling, achieving as well as they should be, as they could, we'll have additional tutorials. One member of my teaching team is very, very good at that and arranges small group sessions where she works together with a small set of students through specific problems. In addition, we have optional tutorials every week. That's mostly me then, where we go through set questions that, but also can discuss anything that students ask at any time. So yes, there is, I think, probably more in-course support and help than in many other master's programs.
Questions in our Q&A box. One of them is, are there research assistant job opportunities?
Yeah, I knew that question would come. The long answer is there may be. The short answer is no. During your program, during the course, you won't have much time. Most of you will come here on some visitor's visa, which actually will restrict your ability to work for money anyway, to a maximum of, I don't know, 20 hours, I think, per week. But working for that length of time is not good for your studies. I personally would recommend you to try to keep your paid work to at most 8 to 10 hours a week at the most. Research assistant jobs are usually only given out to graduates who are able to work full-time. So during the studies, I would expect it to be virtually impossible to find a job that involves actual research. Having said that, I know that this year, and that's new to me, this year one or two of our students, our class, managed to actually get jobs as demonstrators for undergraduate practicals. That's usually, these demonstrator jobs are usually given to PhD students in the college. So I don't quite know how that happened, but it's clearly possible that our students, because you guys have a degree, so you'll be employable as graduate teaching assistant. But I'd be suspicious if there are very many jobs like this available.
Thank you. And then there's another question. Does every student publish a paper associated with their dissertation?
Very rarely. It happens occasionally that a student will then be able to publish a paper. One of our students from 3 years ago did a systematic review type project, and that was published together with the supervisor. Some students who work on research projects that are embedded within a bigger research group, sometimes their work leads on to publishable results. But on the whole, this is not a particularly common occurrence.
Perfect. And there's another question there, which I think you kind of touched on with the working situation, is it possible for a student in the programme to pursue an additional module of interest?
Okay, I asked about that because I didn't quite know the answer. In principle, it is possible to take additional courses, but only if the relevant course coordinator agrees to that. Each course at the university, or in our master's or in other master's programs, has a course coordinator. So this person will have to agree to an extra student to join in. What I would expect, though, is that you would be only allowed to listen to lectures, stuff like that, because we are all very stretched. So adding assessment to this is probably not something that course coordinators would be happy to take on, that they would actually allow you to sit exams in the course or hand in coursework. In addition to that, I should say that if you want to study a particular course and get the credits for it, then that costs extra money. So studying extra courses with the aim to gain extra credits is going to cost extra. Now, I should also say, and I touched on this already, the timetable, certainly in the first semester, is very busy. It doesn't sound like it, I guess, if you have only 3 or maybe 4 days a week scheduled classes. You have a lot of stuff to do. So I think realistically, it is very unlikely that you'll have the time to study or join another course. This is something that really, really good students can do. Those who find it easy to take on take on board the stuff that they have to study and have simply mental capacity and time to study more stuff. So I wouldn't expect that to be possible for many of you.
Thank you. That is us now at the end of our session. That was very fast, and you've managed to cover quite a lot of information. Just before we sign off, is there anything that you would like to add before we go?
I think what we haven't touched on, because I think that was on the set of questions that I've originally been sent, was how do I prepare myself for this master's program, right? Is there something I can do? Now, I promised you I would get back to this. So this is a screenshot from our virtual learning environment called Moodle. And this is a course from the current academic year called Molecular Biology Revision. This is a course, any student who registered on our master's program and matriculates will get enrolled on this course, and that will happen by the end of August, early September, so well before the actual teaching starts, because we have the experience that students, we can't properly judge the background of overseas students. So there are students who have maybe long forgotten their molecular biology or never really learned enough molecular biology. So this is a course where you are able to revise some very basic molecular biology concepts. We're using this genetic disease phenylketonuria as an example, but it's overall more about the basics of what is DNA, what is RNA, what is transcription, what is gene expression, what are mutations and variants. So that's something which you will get access to, and you can work through that, and you should work through this before coming to actually start studying with us. The other thing that you can do is look at this thing here. And I don't know if Naomi can put the URL into some sort of chat box so that you can look at it. This is a booklet called The Genetic Basis of Disease, which is co-authored. So Leah here and myself, we are still members of that teaching team. Maria is my predecessor as program director. Joanna here is in a different school in our college. We wrote this for the Biochemical Society in the UK. This is a free booklet, and it's based very much on our first semester teaching. This is aiming at final year school students, really A-level students, or first year undergraduate students in the biomolecular top field. I think that is a good way of getting into the swing of things and getting an idea of what we're gonna do. There is no real reading list as such. Have you taken that down, Naomi? There's no real reading list as such for our master's program. You will be expected to read and identify published sources yourself and read and evaluate them in your own time. That's about it, what I had prepared here.
Perfect. Well, thanks very much for taking time out of your busy day to talk to us all. We hope you've all found that useful. And like I said, if you have any further questions, you can contact the programme administration teams. So thanks again, everybody, and we hope to see you all in September. Thank you. Bye-bye.
Thank you. Bye.